have this Wasserstein distance, and this sort of distance gives you a very smooth gradient than as if you compare it with the original GAN, and it changes very smoothly. So the advantage is like, it doesn't have any abrupt changes. And if the gradients does not have abrupt changes, then it's actually helps you in making the model much more stable. So the WGAN, basically the quantum WGAN model, it actually outputs a score, like how you get a score for your exams. It's something like that. So even the quantum Weserstein GAN model, QWGAN, it also outputs a score which we call like a critic. Uh, and this output is given by the discriminator between the real samples. So again, the real data points and the generated samples, and then the Wasserstein tries to measure the gap, that how much work needs to be done to to push these generated sample distribution towards the real dataset distribution. So the real dataset has a distribution. You are generating samples. You are generating what samples? Molecular structures. The real dataset also has a lot of molecular structures, which are the true data, and now you are generating fake data, and now you want to compare with the help of Wasserstein, how far can you move this fake data generated by your generator towards the true data, which is your data set. And then in order to stabilize the training, we implement the Lipschitz behavior, which tells you how fast this function can change, and this is in the form of a gradient penalty. So if you see this third equation, if you see here, you have a lambda as a parameter. So lambda is more like a regularization parameter, which we also study in our ablation study. I'm going to show you, and you can select lambda between 0 and 10 or any other number and see how the QGAN performs. And the performance is measured by checking the Wasserstein distance, whether the Wasserstein distance is closing, means it's reducing or not. If it's increasing, that means the generated samples, they are going far from the distribution of the real samples, which means the generator is giving you more fake outcomes. It's not able to really mimic the true data set. And then gradient penalty is a way to control the behavior and then see the performance of the model. So as I mentioned you, so we have a patched parameterized quantum circuits. And this is one of the structures that we use in our research. It's a very standard sort of parameterized quantum circuit. So, Dr. Aswath might have already covered these concepts. But the thing is, in a parameterized quantum circuits, um, you have gates which can be modified by the user. So essentially, rotation gates where you can put your data, you can encode your data as rotations in a block sphere. So that's what we use. So you can use RY, RZ, RX, any sort of gates. And then readout has been already explained. So I'm not going to go into the quantum computing basics because I believe that has already been covered today. But you should be more familiar with this circuit, and this is the C naught gate which you have in between. So C naught gate is used to create entangling operations between the qubits, and that gives them that's how quantum computing is essentially different from classical, because entanglement is something which you cannot really do on a classical computer but only on a quantum computer. So you can of course simulate, but there are, of course, I mean, I'm not going to go into that, but there are entanglement as a resource limitations and so on. You can simulate with classical but only up to a certain point, but you cannot really do an entanglement with the classical. um, so we use these circuits. So this is important to note that this is like one layer. So if you see, if you see my arrow, it starts from this get zero, all the states, then it goes through all these gates, RY, RZ, C naught, etc. And then just before measurement, that's your one layer. Okay, this is like a one layer of PQC. Okay. And then what we do is we make patches of these. So patches means So we basically divide this into two parts, so like two qubits. So only like the first and second qubits, and that's called as one patch. Then if you have like another two qubit set, that's another patch, and then there can be another one, patch 3, patch 4. But each can have like this sort of a layer of like, ry applied, rz applied, then c naught get applied. So you essentially like, have these patches which are like each of like two qubits, but then like, that you can call it as the breadth of the circuit, like, because it increases like this, increase in the number of qubits, so like two qubits, two qubits, two qubits. And then you also have the depth of the circuit, which is like, which you are seeing horizontally. So vertically you have this breadth, and then horizontally you have the depth of the circuit, where you have these layers, ry gates, rz gates, ry gates again, and then c naught gate, and so on. So that's the difference between a patch and a layer, because this is going to come up. So because our circuit is based on four patches, the most optimal circuit that we have got is having four patches and two layers. So two layers means this ry, rz, ry, this c naught, and this structure is repeated again. So again, like, here you can imagine just before measurement, again ry, rz, ry, then c naught is repeated. So that's a layer. Um, so here, like, the three architectures, very quickly. So this is the architecture in the middle. If you see, like, we have the uh, drug fragment information. We give it to a parameterized quantum circuit, and then, like, it predicts some graph structures. They are nothing but molecular structures, as I have already explained to you. And then if you see real and fake, so real data comes from a data set. Here we have used QM9, as I mentioned you, and the fake comes from the generator, which you now know, and it's fed into the discriminator, and then you compute different sorts of matrix. So this is like a standard model which was uh, used before by IBM. Then what we do is that, like, we have applied Wasserstein distance to this sort of model, and then Wasserstein with gradient penalty. So in the model which IBM developed, if I remember, they did not have gradient penalty, but we wanted to apply gradient penalty for better results, so that your gradient doesn't, like, vanishes at a certain point, or it becomes zero when you are training. So, or too large gradients, like, too large gradients makes the training infeasible. So like you basically regularize, it's a regularization parameter, as you saw lambda in gradient penalty. So three models, and that's what we do. So we use the Wallerstein, which I mentioned to you. It helps you smooth the gradients for distribution matching, two distributions generated and real. This is a hybrid template, but you can add gradient penalty, and you can stabilize this with Lipschitz control, which I've already explained. And you can have Wasserstein and gradient penalty together. And that's the best model that we have received in our work. So quantum generator, then classical atom and bond decoding, and then you get the molecular graph. Then you convert that into a classical critic with the help of discriminator, and then RDKit is the library which helps us measure the different chemistry matrix, as you're gonna see. So I think I have explained the architectures. So I'm going to just skip past very quickly. So this is the quantum W GAN, so quantum Wasserstein GAN with no gradient penalty. So if you see, there is like no gradient penalty here on the right hand side. If you focus on the right hand side, because the left hand side is the same kind of model, like, generator with a patched layered quantum circuit, which I have already explained to you now. It generates the graph embeddings, the molecular structures, and then you feed it to discriminator, and then, like, the discriminator is like a critic. It gives a score, as I said. So this is Wasserstein without gradient penalty. um, and then this is the one which was done by the IBM team some time back. Uh, but I think they didn't use gradient penalty, but we have used gradient penalty to their model as well. And and this is more about regularization, as I mentioned you. And this is the model, like, which is our reported model. So if you remember, I was saying you a couple of minutes back that P4L2, so 4 patches and 2 layers. So each patch is of 2 qubits. Again, go vertically, 2 qubits, 2 qubits, 2 qubits, 2 qubits, 4 patches, two, 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 two. And then with each patch, so for this 2 qubit on the top, it has 2 layers. So again, if you remember that layer which I showed you, RZ, RY, that structure, it repeats for 2 times for each of these patches. Okay. So I hope that that's clear. But like, in the end, we can take questions if there are any doubts. But for that model, 4 patches and like each patch having 2 layers, we do the Wasserstein and gradient penalty, and that's the model which we have got as the best outcome. So I think this is just an explanation of the patches and layers, but I think I have covered it. So more like we try with 2 different data sets, um, QM9 and Sync 20. And these are some of the optimization parameters, the training hyperparameters which we have used. So if you see, like, patches are here, 1, 2, 4, layers are also there, 1, 2, 3, 4. And even gradient penalty, lambda GP is also there. Can you see? We have set it to 10. So why we set it to 10, that reason also I'm going to give you. Um, and yeah, and then other parameters are basically the LR is the learning rate. Okay, like, which is a standard parameter in machine learning, because ultimately GANs are just some machine learning models. They just learn from data, though they generate data, but they deal with data, and like, they always have a learning rate, which is again a hyperparameter. You want the learning rate to be either slower or faster depending on your performance of the model, and it depends for each and every individual on what sort of problem they are working on. So yeah. And then sorry, and the batch size is like, how many molecular samples are taken at a time, because if you remember, like, these are all distributions, and you cannot have one data point and make a distribution. If you want to make a distribution, you should have several points. So here you can imagine, like, 64 molecular samples are taken at a time for training. So these are some of the questions that we ask, like how sensitive is gradient penalty lambda, then does the hybrid model match QM9 better than MolGAN, and some other questions, which I covered before. So let's go to the results. Now what we do is that, like, we compare the Fréchet distance model. So Fréchet distance is nothing but, like, having assuming the distributions to be Gaussian in nature, and then just, like, having a comparison between them, how close the distributions are. And Wasserstein is more about, like, how much work you need to do to, like, move this generated distribution towards the uh, the real training distributions. It's about movement. So Wallerstein is about movement. That's why it's also called Earth movers distance parameter. If you check any statistics books, you will find this information. But it also called Earth movers because it's a transport based, whereas Fréchet is more about, like, having the feature distributions as Gaussians, and then you compare between those Gaussian probability distributions. So that's the difference. Um, so we actually see, like, so I think this is pretty straightforward from the graphs, and anyone can deduce, like, what's happening in the graphs. But if you see, if the graph is going lower, that means, like, the distributions are going to slowly match each other, which is good, which is what we want. You know, we want our distances to reduce as much as possible, because that means, like, you are having your generated distribution, the generator, the fake one, is slowly becoming like real. And the major example which you see today, this happens in AI, right? Like if you see the videos which are generated by, like, ChatGPT, Sora, or maybe Google Google Gemini's Nano Banana Pro model, or anything, like, they are, or even ByteDance, like, there are so many different models now, and they all generate videos which are so realistic. How do they work? GAN. GAN is the fundamental behind them. It's the generator which keeps generating those image samples. So here we are generating molecular samples. There you generate image samples. And from image samples, you basically create a collection of frames, because a video is nothing, it's just collection of frames moving very fast per second. So that's why you generate a lot of these frames. But, like, it depends on your training data, like your true data, like, how well you have trained the model. And the more well you train the model, the more realistic the outcomes look. That's why sometimes it becomes very difficult, for people to identify what's AI and what is not AI, which means that the generator is becoming so much capable in fooling the discriminator. You can consider yourself as the discriminator, who is a critic. You can give a score. But let's say you see two videos and you are not able to understand that which one is real and which one is fake. That means generator is successful in fooling you. So same concept. It's the same concept we are applying, but for molecular samples. But it's the same fundamental. And if the distance is reducing, that means, like, the distributions are matching closer. That means, like, you can no longer distinguish between the fake and the real. So the fake is becoming real. Um, and these are just some numbers. So there is a large table in my appendix. I can show you in the end, but this is just a small version of that table, for different parameters which we check for drug candidates. So that will make a bit more sense when I'm going to show you later, like what parameters we are taking. But essentially, like, we are taking, like, quantitative estimate of drug likeness, which collects a lot of different molecular factors and biological factors into account to predict, like, that how your drug is your drug real or not, like, is it really drug like or not. And log p is the octanol coefficient, which tells you about solubility. And then there is a drug candidature score, where you really judge based on all the parameters that whether this is a suitable drug candidate or not, which means, can you use this for, like, synthesis? Is it valid? So, like, valid score. So valid score is, it's very, like, intuitive, right? You can understand valid score means, like, it's going to check the validity, whether it can work in a real setting or not. Unique score means, like, that whether the generated drug samples from your model QW can, with GP, of course, we are now on the best point, GP. So QW can with GP. Um, is it gonna, like, is it new? Essentially, it's unique. Is it new? Like, is it something which does not already exist in your data set, QM9 or either zinc, any of them? Like, is it something entirely new? So that's what you're checking with unique score. Um, uh, One small clarification. So HG, you might be wondering what is HG. So QW GAN is quantum Wallerstein GAN. HG is hybrid generator. So hybrid generator means, like, it's just like, as I said, you hybrid combination of classical and quantum. Quantum is generator, discriminator is classical. So that's why we name HG, hybrid generator. And GP is gradient penalty. As you know, P4 is 4 patches, 2, 2, 2, 2, 2 qubit, 2 qubit, like that. And L2 is layers. Um, so if you see, like, so we compare, like, with molgan, which is a classical, like, molecular GAN, basically, not quantum, but molecular GAN. And we get some results which are, like, which are nice as compared to the molecular GAN at some aspects, if you see. um, and and so here, like, it looks good, that okay, like, so we are happy that, oh, like, quantum WGAN with the gradient penalty is, like, performing very nicely. But then, the reality strikes, and we are going to go to that. It's not always rosy all the time. But for now, like if we see like some of the different validity matrix, whether it's a valid drug, whether it's unique, new, whether it's quantitative estimate of drug likeness QED is higher or not, whether it's log p octanol coefficient is nice or not. The log p octanol coefficient is just solubility, whether you can dissolve the drug in water, essentially. So you might have taken medicines in your life. Some dissolve, some do not dissolve. The ones which do not dissolve, you have to essentially swallow them. But the ones which can dissolve, you can actually chew them, or you can just keep it like in your mouth for some time and they are going to dissolve. Um, but mostly for capsules, if you see, they are also going to dissolve, but not in your mouth. They dissolve with the acids present in your stomach. So, like that sort of thing, like, it's if it's going to dissolve or not, these are the matrix which actually tells you. Um, so I think this slide is about, again, like, comparing the classical and the one which I have already covered this one. And I have also covered that we run ZIG20 as well. Um, it's a more diverse chemistry data set, and this is just for a scalability sanity check, not like a proper state of the art benchmark. But it just gives you an idea, like how your model is performing, because you want to try with different data sets of same sort of problem. Like QM9 has a lot of molecules, ZIG20 has better molecules. So you want to try different data sets and check whether my model is really good or not. Um, and so why do we settle at, so this was a question which I was trying to answer before. So why do we set at 10? So basically in our paper, we optimize for a balance of stability, speed, and drug likeness, so QED, and the lowest threshold distance. So when we like approximate these distributions as Gaussians, how close are they? So that's threshold distance. um, and so we found that, like if we under regularize, so if we have the lambda value as 0.1, because lambda 1 is gradient penalty, right? So if you put a very less gradient penalty, then the training becomes unstable and it's harder to get a nicer value. So if you see, like the threshold distance, the Wasserstein distance, the stability, the for iterations, for validity, QED, and the drug likeness, all these scores for 0.1, they are not really so nice. Uh, but if you see, for gradient penalty 10, we also try between 50 and 100. In like it's not like that everything improves. If you see, like you're at 50, only one improved, which is like Wasserstein distance, but the others are not really improving. Um, and again, same for 100. But at 10, we can see multiple of them actually improve. The drug likeness has improved, the validity has improved, the stability has improved, and so on, and the threshold distance as well. And the Wallerstein distance is not too bad. It's okay. So that's why, like we chose lambda is equal to 10 as our default for the experiments that we do. And again, like for ablation studies, where we have compared, like different patches. Take patch 1, layer 1. We take patch 1 with layer 2. So patch 1 means like only like a single 2-qubit circuit, only one. Okay, patch 2 means 2-qubit, 2-qubit, two of these circuits. Okay, but with different layers. So we found that, like with P2L2, we actually get a better threshold distance, but we went ahead with using P4L2, like this one. And you might ask, why do you want to do that, like when P2L2 is giving you better matrix? So that's just because of scalability, because we wanted to check if multiple of these quantum circuits with two layers, like how much stable they can be, how much scalable they can be, how much can we extend them, and how do they perform with noise. So that's one of the things, like what we understood is, like that if you have more patches or a lot of layers, that does not mean that it automatically becomes better. So that's one insight which you can take from this. Then we go on to noise analysis. As I mentioned to you in the very beginning, we didn't ran on a real hardware due to some constraints, but we simulate the noise. We simulate the noise with some percentages like 0, 1, like a percentage of noise. And for each noise, we simulate, we just measure our models. So for depolarizing noise, you basically have just random Pauli errors, like Pauli X gate error or Pauli Z gate error. Then for amplitude damping, basically it's about like the strength of your qubit, the amplitude of your qubit, like how it decoheres, like how it slowly starts decaying. And same for phase, phase is a very important information for a qubit. That is measured by T2, and we call it like dephasing. So phase damping means dephasing, so how the phase deteriorates over time. For a realistic device, we usually have like 50% depolarizing, 30% amplitude decaying, and 20% phase decaying. And we take those parameters and then we simulate a real noise like simulator. And these are the results. So what do we do? So we checked for quantum GAN with gradient penalty, which has freshet distance. And the baseline is shown in dotted. If you see, it's at the left hand side towards 0. And we see that actually with the noise strength, like as we increase the percentage of noise from 0 to 10, our model performs very poorly, which is kind of expected, which is what you would expect. How do you know that it's performing poorly? Again, pressure distance. What is pressure distance? Just having your distributions like Gaussian, and then measuring the distance between them. And as I mentioned to you, measuring the probability distributions, and if it's getting closer and closer, that's what you want. But if you see it's going farther and farther, if you see this is like around 24 or 23, it's around here, right? And then if you see this is the lowest one, so we want lower distances, both for Freshet and Ballstein. This is true, right? Because that shows you that yeah, you are really getting close to the generator samples are really getting closer to real life data. So that's what we show for both the models. And this was the best model, as I mentioned, the QWGAN with gradient penalty and 4 patches and 2 layers. um, so the patched best model is baseline, and there is like less than 6% degradation is what we observe here. And this is just the same information, but in a tabular fashion. So so those are some numbers with noise analysis. Now let's go to the interesting part. So far we saw that, okay, this model is performing nicely. We can see some quantitative estimate of drug likeness improving, validity improving, solubility, which is log p, that's improving. Okay, uniqueness, okay, somewhat good. But what about the chemistry? But what about the real life chemistry? Are these generated molecules really serve the purpose? Well, spoilers, not quite. And why? So we actually do a lot of analysis on that. So first analysis is admit analysis, where you have absorption, like how the molecule gets into your body and cross relevant barriers, because your cells also have barriers, right? And then you need to cross those barriers. Um, and the distribution, how it spreads through the tissues of your body, then how metabolism take place, how the cell is able to chemically process that drug in your body. So drug is nothing but, you can consider it like a medicine. You're taking a medicine and you expect the medicine to work. So let's say you have fever and you're taking medicine of fever, so you want the drug, the fever medicine, to work. So it depends on these real life factors, right? How the fever medicine can get absorbed in your body, how can it distribute, how can it spread and go into the correct location, how is your body's metabolism, how are the chemical processes happening in your body, how is the excretion being done, what is the toxicity level of that drug, is it harmful, does it have harmful effects? So like for any medicine, any drug taken too much always have some toxicity. harmful effects because you cross a certain threshold and then it becomes toxic. So so the first is like, about log P. So I think log P I have already mentioned is just water solubility. And if you have more score, that means like, you are going closer towards solubility. And I think from the graphs, if you start checking the graphs, then you will see that, it's like the model is like, performing not really good for some of them, for the molecular weight, MW especially. If you see like, your real is denoted in blue, that's the blue distribution. Okay, and this is what I mean by distribution, by the way, if you were wondering what is distribution. So something which looks like this, these are like, just collected samples and like, how they vary. So that's your distribution. Um, Essentially like a probability distribution. So, and the red one is generated. So if you see for log P, it's actually good. It's not so bad, right? Because it's like, if you see the red one, it's very close and overlapping with the blue. That's what we want, right? Like we want the red one to be very close and overlapping. So that's what I mean, right? By Weatherstein, Freshet, all these things, right? That's what we're trying to get to. Closer and more overlapping distributions. So if you see for molecular weight, which is nothing but like, it's just like, how heavy your molecule is, literally. So it's so if you have like two small molecules, then like, there are chances of getting it into fragments. So like they break apart, basically they break into fragments. But then like, if you have two heavy molecules, then that is kind of hard for your body to digest, right? Because then the permeability threshold is not crossed. So it's not really absorbed by your body if it's too heavy. And if you see like, our model is unfortunately not performing well in that parameter, which is molecular weight. But in TPSA, which is like the polar surface area, okay, which is about like, which tells you about like, how your intestines uh, like, are permeable, like, the permeability of intestines in your stomach. If you have like TPSA like too high, then you will of course have like a lower permeability, right? Like, because intestine will not be able to like, really absorb and like, take it into the bloodstream. But then if it's too low, then like, then it's not soluble at all. So on one extreme you have like, not soluble, and in another extreme you have like intestinal permeability, which is a fundamental parameter here. So you want your TPSA also, like, but for the TPSA, like, it's not too bad. It's like you have the distribution, the real from QM9, everything is from QM9, and the red one is, yeah, it's overlapping. If you see like, you can see the so the dotted ones, so the dotted ones are basically, it's like, the center of mass of the distribution, basically the dotted lines, the blue and the red dotted lines. So if you see like, for TPSA, the center of mass is lying somewhere towards um, in the real data distribution. So it's somewhat close. Again, not totally nice, but somewhat okay. You can say this is an okayish result. But then QED, quantitative estimate of drug likeness, which we have already seen, the average is low. So if you see like it does not really overlaps at all. Like, I mean, the main peak, uh, and even the center of mass, the red dotted line, even that also doesn't really overlaps with the real distribution. So which means like, we have a like, smaller like, higher QED cluster. So that means the cluster of molecules, the distribution, like, it has a very smaller score of quantitative estimate of drug likeness. That means, which means like, estimate of drug likeness. So what does it mean? It means that like, it's not totally a real estimate of drug likeness. The score is lower. You want higher marks, right? In order to success, we want high scores in subjects in order to pass. So it's like that. You have lower marks here. You can consider the score as a marks, basically. You're giving marks essentially. Um, Same thing we see with the drug likeness score as well. That's in the, but we took a composite drug likeness. So we take QED, SA. SA is synthetic accessibility. So synthetic accessibility is something like, if you can like, if you can, so in simple words, if you can make this in a lab or not. That's it. Can you make this in a lab? Can you make this chemical in a lab? That's the thing. That is SA. And there are other parameters like Lipinski. So TPS I have already explained. Toxicity I've already explained. HERG we will see later, but HERG is something related to heart. It's something related to cardiac. Okay. And Lipinski rule of five, which tells you oral drug likeness. So can you take a medicine orally, which means that can you keep it in your mouth for some time, and your mouth does not start to swell or any other bad reaction happens. That's Lipinski RO5 rule of five. Um, and I'm explaining to you in a much more simpler words, but there are a lot of other nuances. But if you see, that also is not totally very strong in our model, and you will be able to see that in the table, because if you see, some of the targets are not actually met. So if you see the generated, now you can actually compare the numbers with the generated and the real data set. So this is what I have been talking about all along. And now for this analysis, it is extremely clear to you immediately by checking the numbers. If you see, and what is the target? So I've also mentioned the target, what you should be having, but what you do not have. So for, if you see, the true data set has a 122.1 uh, molecular weight, but our generated is only 40.2. So not good at all. The target should be between 150 or 500. So even for QM9, if you see, it's a little bit below the target, but still very close to the target, very close to 150. So far better than what we have generated. Same for log P. So what's the point of this? You may ask that, oh, the model is not performing good in real world matrix. So then what's the advantage of having this kind of model? To be honest, no advantage. But that's the part of research. Research means you explore, you investigate, you come up with new models, you investigate whether they work or not. They are going to work on part of research, and that's what I'm trying to show you here, how a real research is done on a real problem, which is drug discovery, which is one of the most difficult problems right now for pharma companies, because of the number of years of research and the money it requires. So if we can have a quantum model which can actually improve on that, then that would be really good. Okay, so if you see, for the target, we should have a RO5 pass of oral drug likeness should be about 100%. Can you imagine? That should be 100%. But our model passes only 25%. But QM9 passes 99.4%. So very impressive. So in this way, I have shared the slides with Dr. Katyayani, and she's going to share with everyone. You can take a look at these table and identify, and maybe come up with something else, because there is a lot of cutting edge research going on, which I'm going to discuss later. And these are the same Lipinski rule of five in terms of graphs. And there is toxicity analysis as well. So if you see, the generated is the average alerts per molecule, structural alerts is much more higher, which means the molecule is not really closer to the real world, the QM9 data set. Um, we also take into account the mutagenicity, which basically it just tells you about the structural warnings. If it can be DNA reactive, if it can cause DNA mutations, like you have in cancer, in cancer, some deadly cancers, they actually mutate your cells, and even they can react with the DNA, which is your core, like you know DNA is the main fundamental acid, which is responsible for your life. So and mutating DNA is extremely dangerous for your life. So that's why, we check the drug from all angles. You can see, we are covering all sorts of things. Can it dissolve in water? Can it be taken orally? Okay, can it dissolve in the body or not? Everything. Can it cause cancer or not? Can it mutate DNA or not? Can it have a heart risk, heart disease, or so can it cause heart disease in the long term? So we are taking into everything into account. If you see, for a real drug, that's why That's why this problem is very hard. You take into account so many different sorts of parameters. So why it's hard to develop a drug which is very optimized. And that's why it takes tens of years of research to get these things into picture. Um, and then next, so this was the ADMET analysis. Then next we do a scaffold analysis. So scaffold is just like a scaffold, like you have a core molecular structure, like let's say the ring system, which I showed you, the benzene ring. And there are other molecules which are connected to that ring. They are more like side chains to the main core. So what we do is we create a molecular scaffold. So we remove the side links with the decomposition algorithm, which I've shown you here, Bemis-Markov scaffold decomposition. So what that algorithm does is it removes these side chains, but it preserves the core framework, the core structure, and the connectors and the connecting linkers. And Yeah, so essentially what we have is the main framework of this molecule. And then we convert each of these into standard smiles representations. Okay. So smiles representations is also what is smiles? So smiles is basically it's a way of writing the molecules as a text so that you can feed it into your ML models or whatever other models you are training. Okay. So let's say in a text, so let me give you an example. So let's say carbon is written as C, nitrogen is written as N, and maybe the bond between them can be written by a dash. So using texts, basically using texts to denote, to represent the molecular structure. Because then with that sort of representation, when you're able to denote it with text, then you can feed it into your model easily. Uh, we do so for each of these scaffolds now we also measure the average size, in terms of like heavy atoms, so basically how heavy these molecules are, and how many and of course with many atoms, if you have let's say like you know zero like heavy atoms, then that means you do not really have a molecular framework like a core structure. So and you do not want that, of course. Like you do not want a scaffold with zero heavy atoms. You at least want some atoms, right? So the observation that we made is for this scaffold, there were 0%, so there were no new scaffolds at all. So all the molecules, the structures which were generated by the QWGAN, gradient independent HEP, no, like 0%. Like there was no new scaffold at all. You would say, okay, this is very bad. I said that, yes, of course. Very honestly, this is bad. But again, as I said to you, it's research. You have to do the analysis. Until and unless you do the analysis, you would never know like what how your model is actually performing in real world. So it is 0%. So it's not good. It's not good. Of course, it's not good. And this is an unhappy result. But of course, it's a result, whatever that is, whether happy or unhappy, we don't care at this point. It's a result. The point which you should be talking about is how can we improve from here. That's the point we should be talking about. So if you see only 30% of these generated molecules actually they are falling inside our paper's drug like qualities, the log p, which I mentioned you, the solubility and the topological polar surface area, TPS limit. Whereas for QM9, it's actually 80%. So QM9 is very good. But of course, it has to be good because that's our true data set. If the true data set is bad, then your generator will be always throwing bad samples, which means the quality, and this is a very important point, the quality of your training data should be absolutely high and unbiased. That's the most important message. Um, so yeah, so I think so what we find is that the outputs collapse onto a degenerate scaffold family. So there is like a same looking molecular structure like degenerate, which are like same level molecular structure family, and therefore it's not able to build meaningful frameworks. So that's the meaning of the scaffold being generated and not very effective. So essentially, we do not have The generated structures, they do not have a core framework to be drug-like or to support solubility, log P coefficient or TPSA. Then next we do fingerprint analysis where it's essentially again, it's like a sort of an encoding of a molecular structure into a length and usually in binary terms. So instead of you storing the molecule as coordinates of graph, which we were doing, of course, you want to know whether there are any structural patterns because you want to learn about the structures. So you want to see if you can really get something from the structural pattern. So here the molecular footprint is nothing but a structural pattern. And we do that analysis as well. And if you see the green ones are generated. Um, but if you see they do not really overlap with the approved drugs by ChemBL. So ChemBL is a data set which has properties for drugs which are approved by different institutions, by different companies, which are approved means which is possible to give in a human body or even in an animal. So, but if you see our graph again, we are facing a problem here. It's not really, only very small part is really overlapping with the approved drug matrix, the ChemBL database, but most of it is actually not. So which means so even for fingerprint diversity and the similarity, we are not doing too well. So we are not doing too well for scaffold, we are not doing too well for fingerprint diversity. For admit, we were doing okayish, I would say, not too good. And then finally we do a docking, so which means you take a candidate molecule and then you computationally test if it can really fit into a binding site. So that the means of, so docking, how ships dock at the dockyard, so something like that. And docking is very important. Why? Because let's say your drug has a structure, a molecular structure, and you have proteins in your body. So would that structure bind well with the protein in your body? That's docking. So what we did was that we generated 100 molecules, the best performing ones, by the way, from these, from what our model gave us, and also 100 molecules from the classical GAN model, not quantum, so the classical one. And then the purpose was we wanted to check the suitability for structure-based screening. So docking ready analysis basically gives you the screening. And for the smiles, if you remember, I mentioned you the simplified molecular input line entry system, which is if you have carbon, you say C, and if you have a bond, you can say a dash, and if you have a dot, that means the components are disconnected. So C dot N means carbon is not connected to nitrogen, but if C dash N is, there is a bond. So so if you have C dot N, that means they are just fragments. So it's carbon fragment or nitrogen fragment and so on. So and we saw that very minor, very small outputs were actually fully bonded. The especially for the molecules that were generated by our model, the structures, uh, even including simple ones like the alkenes and the nitrile bearing, the nitrogens, and even they even the simple ones they were not connected with each other, they were fragmented. um, so which means they are not optimized for drugs at this point. And this is what we see. So if you see, this is a representative string, so where so 60% of those molecular, the molecular structures out of 100,
thank you
60%.
thank you so much, Srinjoy. It's very innovative to talk to you, because of in our YSR engineering students having the subject, same subject, what you are saying, the session, it is very useful for us. Thank you so much for your kind cooperation, and we have the limited time we are giving to you. but you are agreeing with our invitation and accepting our timings. Thank you a lot. We are grateful to you. Any queries on online or offline, please ask freely. Please in chat and web. Otherwise, please chat in the chat box. Post in chat box also, no problem.
Thank you. Thank you so much. I'm glad that it's very helpful. So actually, I'm also sorry. Yeah, sorry. I was just saying that I am actually done. So, sorry, I can't hear you. Like,
Go ahead, Srinjoy. Now, go ahead.
yeah. So what I was saying is that like I am done. So most of the major part is done, which I have explained to you. This is just discussion slides, which were like the things which I've been telling you through the presentations, that we have worked on a noisy model. We have a hybrid adversarial training. We have better diversity than Molgan, but again, at the comprehensive chemistry diagnostics, we fail. Uh, Definitely for scaffold novelty and fingerprints, for the docking as well. And we did somewhat okay for admit analysis, but again, like okay, it does not works because in real world you need to be really sure because you're going to use these drugs on either animals or humans. So yeah, so and then like I just wanted to say about some future scope very quickly. So what you need, like so since you saw that like the chemistry performance is really not good with the model, that means you need to have more chemistry aware generation so that the generator knows and like corrects itself in real time, that okay, like this sort of parameter like scaffold is not giving like the generated structures are not passing the scaffold test, so that means it can regenerate again, so making the generator more aware. So that's one of the goal. Then more sort of optimization is needed for medicinal, by keeping medicinal chemistry constraints in mind. We can do quantum engineering, of course, but again, for quantum engineering, we would like to, then you should be focusing more toward tolerant or like fault tolerant machines. Of course, if you we saw that like it's not always about having like so much breadth or like so much layers, so much depth. So again, two large circuits really does not help. You have to really check like how it's how your model is able to capture the essential information from the structure. And for that, there are different techniques. There are different research which is being done.
And that when we are going to the program, please allow students stand up. National anthem.